Ma'am, should we start today?
Yes, Amrita.
Yes.
We can proceed. Yeah.
Okay, thank you, ma'am. So, good evening. Welcome to this webinar. We have gathered here to celebrate the 159th birth anniversary of the father of Indian chemistry, Acharya Sir Prafullo Chandra Ray. I would request our respected principal madam, Dr. Shoma Ghosh, to inaugurate today's session. Over to you, ma'am.
Thank you, Amrita. It's really wonderful being gathered over here again in your webinar. And as you mentioned that we are here today to pay our tribute to Acharya Sir Prafullo Chandra Ray. As we know that the Royal Society of Chemistry honored his life and work with the first ever chemical landmark outside Europe. And he was the founder of Bengal Chemicals and Pharmaceuticals, which is India's first pharmaceutical company. He is the author of many legendary books and scientific creations. And now, on this auspicious day, we could organize such a webinar. It is really a proud privilege for me to welcome all of you, all the participants, and of course, the speaker, Dr. Sanghita Sinha Roy. And I'm really grateful to Dr. Sinha Roy for sparing some of her valuable time to deliver her lecture in our webinar, and hope that we'll listen and be enlightened by her speech, and will come to know lots about the scientific discoveries and scientific innovations, scientific contributions, especially in relation to today's health crisis. As we know that the world is facing a tremendous health hazard, so hope that Dr. Sinha Roy will enlighten us on this auspicious day, and with my tribute, with my wholehearted respect for this father of Indian chemistry, Acharya Prafullo Chandra Ray, I'd like to request Dr. Amrita Banerjee to proceed further with the webinar. And of course, I am grateful to Amrita and her team in the Department of Chemistry who thought about this on this very day. So let's hand it over to Amrita. Let me hand it over to Amrita madam for further necessary proceeding. Thank you. Over to Amrita.
Thank you, ma'am, so much for your valuable and inspirational words as always. Now, before we move on to today's talk, I would request Atreya or Pooja to play a small video made by the BSc Honors final year students of our college from the Department of Chemistry as a tribute to Acharya.
Acharya Prafullo Chandra Ray, an eminent scholar, educationist, chemist, and a philanthropist, was indeed a man ahead of his time. Born on 2nd August 1861, a Bengali man who changed the course of the medieval chemistry in India with his pioneer work. Undoubtedly, the title given to him as the father of the chemical science in India is justified by his deeds, which not only benefited the chemists then, but also paved way for the future endeavors in the field of pharmaceuticals and drug development. Prafulla Chandra Ray was born in Raruli, Kachipara, now in Bangladesh, in a Bengali family. His father, Harish Chandra Ray, had a great love for learning and had an extensive library in his home. His mother, Bhuvan Mohini Devi, was a compassionate lady with liberal views. He had two brothers. He completed his schooling from Albert School and joined Metropolitan College for further studies. He had profound interest in chemistry, so he joined Presidency College and even built a laboratory at home for experimenting. In 1885, Prafulla Chandra Ray won a scholarship to Edinburgh University, UK, and gained his doctorate degree in 1887. In 1887, he also received a Hope Prize for his thesis on conjugated sulfates of copper magnesium group, a study of isomorphous mixtures and molecular combination. On returning back to Calcutta in 1888, he became an assistant professor of chemistry at the Presidency College. Having established a research laboratory and gathering a group of dedicated students, he was able to publish 150 research papers during his lifetime. Satyendranath Bose, Gyanendranath Mukherjee, and Gyan Chandra Bose were some of his notable students. His research included the discovery of stable compound mercurous nitrite in 1896. He also researched organic compounds containing sulfur, double salts, homomorphism, and fluorination. He even did research work on food adulterations and its consequences. In 1892, with a small capital of rupees 700, he established Bengal Chemical Works, which later on pharmaceutical company in 1901. It is now one of the leading chemical and medicine producers in India. He was a voracious reader, and his interest in the ancient text led him to publish The History of the Hindu Chemist in 1902. He was even elected as the president of the Indian Science Congress in 1920. Due to his notable works, he had won many awards, like Companion of the Order of the Indian Empire in 1912, Knight Bachelor in 1919, Foundation Fellow of the Indian National Institute of Science in India in 1935. His autobiography, Life and Experiences of a Bengali Chemist, has been a notable inspiration since decades. Apart from being a renowned scientist, Prafulla Ray was a passionate and a devoted social worker. He actively participated in lending help to the victims of the flood and famine in Bengal in the early 1920s. He established many industries like Bengal Enamel Work, National Tannery Works, Calcutta Pottery Works, etc. His contribution towards Brahmo Girls' School, Nagarjuna Prize, and Bengal Relief Committee makes him a gem of a person full of empathy and humanism. He persistently carried on his work as a professor till 1936 and died on 16th June 1944 at the age of 82. His life teaches us the lesson of being passionate and hardworking. Though he led a very simple life, but his works made him extraordinary and one of the most influential individuals in
Nisha, Annu, Piyu, Priya and Khushbu for their contributions in making this video voice was of Khushbu Parveen, and obviously Atri and Pooja, who did the final video compilation. So thank you all. So today we are privileged to have Dr. Sanghita Sinha Ray from the University of Texas MD Anderson Cancer Center. Without getting into detailed formal introduction of our speaker, which has already been shared in the brochure, I would say a few words on her research. She pioneered the development of multi-diamagnetic MRI contrast agents and MRI methods that exploit chemical exchange saturation transfer for the detection of enzyme activity within in vivo tumor models of ovarian, colon, and pancreatic cancers during her doctoral research. Her postdoctoral research included evaluation of neuropathology in several infectious diseases such as HIV, cerebral malaria, Ebola virus diseases, etc., using non-invasive imaging methods such as MRI and PET imaging modalities. This young, talented researcher has many awards and has represented in several international conventions. I personally know her for the last 16 years, and she happens to be a very close friend of mine from JU days. So it was very easy for me to get her consent in spite of her very busy schedule. Welcome, Sanghita, over to you.
Thank you, Amrita. That was really a wonderful introduction. And I don't think I deserve that much credit, but I really appreciate all that all you and Dr. Somadas said earlier. And I also enjoyed the presentation that you guys just showed. It was all the other details of Dr. Acharya Prafulla Chandra Rai, and he's truly a man way ahead of his time. He was a man way ahead of his time, not just as an eminent scientist, but his contributions continue to inspire all of us even today. So it is an honor for me to present actually to this great audience today. And with that, I'm going to start sharing my screen and present a little bit of the work that I have been doing. Okay, so good evening, everyone. And today, it's like I mentioned before, it has been a real honor for me to present on this very special occasion some of the research that I have been doing during my PhD and postdoctoral studies towards the advancement of healthcare, the future of healthcare through molecular imaging. As you can see in this slide, molecular imaging is a really rapidly advancing field, but it's really very multidisciplinary. So it involves the real success of this field actually involves a lot of good interplay between various biomedical disciplines. Most of the work that I have done focuses on some of these areas, such as contrast agent development, some imaging physics, some molecular and cell biology work, and some in vivo validations within small animals as well as in rhesus macaques. So this is just the slide as an overlay, overview of my academic career. And after Jadavpur, I transitioned more from, well, my background was in chemistry, but I transitioned from being an organic chemist to being a more translational imaging scientist now. But it's a very wide scope. I feel that chemists can evolve into, so that has been a great journey so far. with that, I'm going to move on to the science. So this is just an overview of, the drug discovery and development pipeline for most drugs in either cancer or infectious diseases. And to kind of give you an idea about these biomedical imaging modalities that we hear about that play a very important role in all of these stages, starting from target identification of the drug to clinical phase trial development. And why is molecular imaging such a big deal, or it's such a rapidly evolving area right now in advancing personalized healthcare? That is because for the longest time, the most common or current practice to evaluate the pathology of the tumor tissue has been to collect a tumor tissue sample and then perform ex vivo biopsies of these tissue samples. Molecular imaging, on the other hand, is unique because it can non-invasively provide to us real-time information about the tumor microenvironment or the disease environment by letting us know in real time what are the changes that are happening in the disease environment as we move on. Now, this real-time information and the fact that it is non-invasive can really help to improve the early detection of the pathology, or to also assess early response to some of these chemotherapies or some of the treatment strategies that we have. Now, as you can see here, mostly, when we go to the clinic, you see we hear about MRI scans and CT scans or ultrasound and PET. I hope to convey in today's talk, as clinicians, clinicians often obtain anatomical information about the diseased tissue when they perform these imaging scans, but before the clinician, not just before the stage, it's also in the research fields with the rapid evolvement with anatomy as well. Along with anatomy, you can use these imaging modalities to actually give you functional information or information about so many of the physiological processes that are occurring in the diseased tissues. So I'm going to mostly talk about MRI and PET, which I've mostly focused on. So if we look at MRI contrast agents, a variety of MRI acquisition protocols have evolved, and they broadly belong to either of the two relaxation methods. It could be either the T1 or longitudinal relaxation methods, or it could be based on the T2 or T2 star relaxation methods. And by this, what happens is when you talk about, for example, T1 weighted MRI or T1 weighted MRI contrast agents, you have agents such as these, which are gadolinium or manganese based agents. I put an example here of a gadolinium chelate agent that is widely used in the clinic over the world for acquiring MRI, T1 weighted MR images. And because gadolinium and manganese agents have unpaired electrons, they can rapidly they have unpaired electrons, so so these tissues can rapidly improve the imaging contrast or change the T1 relaxation times to give you different image contrast between pathological tissue. And here in this picture, before you inject the gadolinium agent, you can't actually differentiate, or you can't see the tumor very clearly. But after the gadolinium agent is injected, it accumulates in the tumor, and because of the T1 relaxation time of this gadolinium agent, you can see that it creates this contrast enhancement, this brighter contrast enhancement that you can then diagnose tumor. So it's a very good diagnostic imaging agent. There is also other agents that are based on T2 or transverse relaxations or T2 star, which basically involved paramagnetic iron oxide or super paramagnetic iron oxide particles. So because iron oxide particles are these super paramagnetic agents, they can create small local magnetic fields that are randomly oriented to the main magnetic field of the MR instruments. And these local magnetic fields then can cause MR frequencies of the water protons to change, either accelerate or decelerate, so you get a different contrast. Has been done with these agents. You can see here, this is a HER2-specific antibody to which this super paramagnetic iron oxide has been designed in such a way that when it binds to this antibody, it gives you an MR contrast by changing the relaxation. And if you look at the image here, you can, I don't know if this slide shows up well, but you can actually see that this is pre-injection, the first slide. And after it is injected over time, because T2 relaxation is the opposite of T1 relaxation, where you're actually seeing a decay of the curve, you can see that the contrast becomes darker in the kidneys. So you can act, and then after 90 minutes, it slowly starts to wash out. So then it again starts to become brighter. But this is how you can again use it for diagnostic imaging or anatomical imaging. But really, MRI has actually evolved beyond anatomical, giving you just anatomical information, as I pointed out. And other MRI methods are being rapidly or emerging right now. One of these methods that I focused on was chemical exchange saturation transfer, or Cest MRI. And Cest MRI contrast agents, they are different from T1 or T2 contrast agents or T2 star contrast agents because they have the slowest chemical exchange rate when you compare to all of them. Now, having the slowest chemical exchange rate obviously brings a limitation that they have the least sensitive detection, meaning that you require a minimum concentration in the single millimolar range to be detected, right? But this is also an advantage because then the slow exchange rate provides opportunities to design agents with specific exchange rates, which provides outstanding diagnostic selectivity. So Cest agents, they're rarely used for anatomical imaging. They're mostly used as responsive agents to detect change in a biomarker. And I'm going to go into biomarkers later on, but biomarker basically means a change in a physiological process or a biological process. So Cest agents can, for example, detect change in enzyme activity or pH or a functional process, basically. And that's very important because some of these functional processes are clinically relevant biomarkers, right? And before I move on to that, let's just go into what Cest MRI actually, or how it functions. To do that, I'm going to just do this through an animation. So Cest MRI agents, they always have an exchangeable proton. To show that, I'm showing an amide proton here. And what happens is when you apply a selective radio frequency pulse, it causes this proton magnetization, or the magnetization on this amide proton to get saturated. When this amide proton then exchanges with a water molecule proton, it transfers that saturation magnetization to that of the water proton, causing a decrease in the signal or the MR signal of the water. And this process keeps going on because you have a lot of water molecules compared to the agent proton, right? So this is again just the animation to kind of show you, you apply the radio frequency pulse, you saturate the magnetization on this proton, then this proton exchanges to the water proton, causes the magnetization to be transferred. And when the magnetization gets transferred, you see a lowering of the water signal that... Sorry? Okay.
You proceed. There is some technical problem.
Okay, thanks. Okay, so just to reiterate that this lowering of the water MR signal, you can monitor with standard MR methods, and that is what we do. So to then show you how the Cest spectrum looks like, this is example I'm using based on a salicylic acid molecule. So this is my raw Cest spectrum. This peak that you see here at 0 ppm with the complete loss of the water signal is from all the bulk water protons that get saturated and have exchanged, so no signal, right? Here at 0 ppm corresponding to the bulk water protons. Then you also see these two peaks here. And this peak is corresponding to the decrease in the water signal due to this salicylic acid proton that was saturated and then exchanged with the water proton, right? So then you can account for changes in the chemical shift of these exchangeable protons on the contrast agent using this MR method. And you can quantify such a Cest spectrum. This is basically a line spectrum. So you can apply line shape fitting protocols such as Lorentzian line shape fitting that I used here to fit this spectrum and quantify the percentage water signal to percentage Cest signal. Saturation frequency for chemists is, equivalent to the chemical shift that you see in an NMR spectrum. So I had 2 peaks. This was from an amide proton that the salicylic acid unit had, and this is the salicylic acid proton that gives you this percentage signal. And you can do, so when I do in vivo MRI, then I apply additional processing that I will discuss a little bit more in detail when I show you an example of that. So going back to that phase where I was talking about all these biomarkers that you can detect, there are a lot of clinically relevant biomarkers that are feasible to be detected with different imaging modalities. Every imaging modality has their pros and cons. Some have limited sensitivity, some have limited resolution that they can provide. So based on which biomarker you're trying to detect, we choose what imaging modality fits, right? So these are the biomarkers that are feasible to be measured with Cest MRI. You can measure enzyme activity, which was the main focus of my PhD research, with agents such as these that change their structure or change a functional group when an enzyme acts on it, basically. You can also identify gene expression or changes in the metabolite levels in the cancer tissues. You can look at environmental markers, which are also signature markers for tumor tissues. We know that tumor tissues are more hypoxic, for example, in nature. You can use Cest MRI to look at hypoxia levels. Currently, I'm focusing on looking at tumor tissue pH. As we know, tumor tissues are very acidic in nature, so that can be also used as a signature to identify early stage tumors. You can detect redox state or changes in the redox state in tumor tissues or changes in the metal ion. There are a lot of biologically relevant metal ions, as we know, and they change oxidation states depending on the disease state, right? So you can also detect that with Cest MRI. So moving on to my PhD project. So the main focus of my PhD research was to design and then to apply these chemical exchange saturation MRI contrast agents to detect this catalytic activity of enzymes in different tumor models. And here are some of the enzymes that I have focused on. I'm going to explain or show you in a little bit more detail one of these projects. And moving on to that first project, this is the one that I looked at detecting gamma-glutamyl transpeptidase enzyme activity. So actually, before I move on to this, I just want to make a side note that one of the main unique prospects of my PhD research was also to kind of see if we can move away from gadolinium-based or metal-based potentially toxic MRI contrast agents to try and design more non-metallic, potentially non-toxic MRI contrast agents. So all the agents that I developed were salicylic acid-based non-metallic agents, okay? So going on, the first one was where I looked at designing a Cest agent to detect GGT enzyme activity. Again, most of the enzymes that I was focusing on were proteases because proteases play a very significant role in tumor growth invasion, and they're also upregulated at very early stages of the tumor. So detecting protease activity in vivo, although it appears a little challenging, it can significantly improve early detection of small primary tumors and therefore provide new strategies for cancer therapy. GGT by itself is a very important enzyme that plays a central role in glutathione metabolism. By cleaving this gamma-glutamyl bond, it actually maintains the cysteine homeostasis. However, because it is upregulated at such high levels in early stages of ovarian and some other cancers, it is a very important biomarker for ovarian cancer. So when I started this project, I came across Dr. Kobayashi's work at the NIH. They were doing this very interesting project with this rhodamine-based optical dye, which got cleaved by GGT to release this to form this fluorescent agent. And the aim of this study was basically, or the aim of this work was to use this agent, therefore, for optical surgical navigation. Now, as many of you might have heard, optical surgical navigation in the recent years have been really surgery, because earlier during standard surgery, as you can see, there are with the naked eye, it's very difficult for a surgeon to identify smaller micrometastases that occurs in the tumor. And so when the larger lump is usually removed, but the smaller micrometastases, because they're not visible by the naked eye, they get missed. And this leads to a recurrence of tumor. And you will often hear that patients undergo surgery, cancer patients, and the tumor recurs somewhere else. It metastasizes, it's somewhere else, it recurs, and it keeps on. So with this method, because it could identify this fluorescent dye could identify very small micrometastases, it's very easy for the surgeons to detect these smaller micrometastases, leading to a more complete resection of the tumor and therefore improving patient survival chances. But the limitation of the study was that only 2/3, since this dye is actually based on enzyme activity, right? So the limitation is only 2/3 of the patients have this active GGT enzyme. So therefore, I proposed that we could use our Cest MRI method to pre-screen patients before they enroll for expensive surgery, to pre-screen them and tell us beforehand which patient has that active enzyme activity and which patient doesn't. So as a side goal of my Cest MRI project, I could use this method to pre-screen the patients for GGT enzyme activity. And moving forward into the main developments in the project, the first step that I did was to design the agent. And as I mentioned before, all my agents, I based it off this framework of 4-amino-salicylic acid. And, the first step is protection of these hydroxyl and carboxylic functional groups with benzyl protections. And then I did a peptide coupling, introduced this protected glutamate moiety, and then remove the BOC and terbutyl ester functional groups with a deprotection with trifluoroacetic acid, and then a final deprotection with hydrogen lysis to remove the benzyl functional groups. And this gave me my final Cest Mri contrast agent that I purified with HPLC so that it could give me a 99 or above % of purity for preclinical studies. So now that I made the agent, I wanted to see whether it gives me the Cest activity or whether it gives me the Cest signal that I wanted to identify. So just to reiterate, we could apply a selective radio frequency pulse to saturate this amide proton. And then this amide proton can exchange readily with the water molecule proton, thus leading to a lowering of the water signal corresponding to the amide chemical shift. And this is exactly what you see here in my raw Cest spectrum of the agent in PBS buffer. You see that I do get the Cest signal at 5 ppm or about 5 ppm that corresponds to this amide proton. And in a similar phenomenon, I also get a Cest signal at 9 ppm that corresponds to the salicylic acid proton exchange. The red line shapes that you see here, they are from the Lorentzian line shape fitting of this spectrum to give me the percentage Cest signal for the two peaks. So this was great and encouraging. And so now let's see what happens when I add the enzyme. So when I add the GGT enzyme to this, what I'm expecting is that this amide proton would get cleaved. So I would lose the amide signal because amine protons exchange so fast that in the Cest MRI scale, you can't detect it. So if you look at the spectrum that I get, yes, I do see the loss of the amide signal. This is shown by the faint blue line here. You can see that I lose the amide signal once I added the enzyme, and the salicylic acid signal remains. So this was a very clever design of an agent, because for the first time, we had this agent that had an enzyme responsive and an enzyme unresponsive signal from the same agent, which means that you could apply a ratiometric approach to get the enzyme activity in a concentration independent manner, which is very important for translational studies, because then you don't have to care about how much of the agent went into the tumor to give me that signal. Okay, So then the next step was to actually check the enzyme activity doing Michaelis-Menten enzyme kinetic study. And Dr. Kobayashi had gifted me or given us very graciously his fluorescent agent to compare with my Cest agent. So these are from the studies I did with his agent. And basically what I did here is I prepared different sample concentrations of my Cest agent. I added a very small concentration, same concentration of the GGT enzyme. I put it into the 70 MR scanner. And these are solution studies. So in the absence of an MRI scanner, you can do this experiment in a 300 megahertz or 400 megahertz NMR spectrometer, because 300 megahertz corresponds to a 7 Tesla magnet. So these experiments can actually be done in an NMR instrument. I chose to do it in the MRI so that I could directly compare with my in vivo results in the animals. So I put all the samples together into the magnet and acquire a 10-hour, so I did an overnight dynamic Cest MRI scan. And what I'm trying to show here in the figure on the very left is just the AMIT signal. If you look at just the AMIT signal at about 4.8 to 5 ppm, you see how it slowly decreases over time, thus telling us about the enzyme activity. This is decrease of the AMIT signal for all the different concentrations that I did. And you could use this graph to get the slope of the first 2 hours, which would give you the initial velocity of the reaction. So I can use that initial velocity to fit it into a Haynes-Wolff kind of plot and get the enzyme kinetics parameters. Now let's focus on the enzyme kinetic parameters, and you see that the Michaelis constant of my agent is about 550 folds higher than that of the fluorescent agent. And what this means is that the Cest agent had a much weaker binding to the enzyme compared to the fluorescent agent. If you look at the turnover number or KCAT, they're almost comparable, but the Cest agent is still half lower, or 0.5 times lower than that of the fluorescent agent. Now, combining the Michaelis constant and the turnover number, you get catalytic efficiency. And the catalytic efficiency was drastically lower. It very concerning. And because what it's telling us is that that GGT might not be able to cleave sufficient amount of the Cest agent during the time frame of the in vivo studies. So it might not work in an in vivo scenario. The efficiency is just too low. So I decided, I still went ahead, and I decided to just, perform in a very small cohort of animals this in vivo study to just see either how inefficient it was, or do I see some signal, or what happens. So I prepared orthotopic tumor models of ovarian cancer. And I chose OVCAR8 as my first model because OVCAR8 showed a very high expression of this GGT enzyme. And this is just in two mice that I studied this. The figure on the very left is anatomical MR images. I acquired the anatomical images in the very beginning to locate the position of the tumor. And once I can locate the position of the tumor, that is when I inject my agent. So after injection of the agent, I wait for around 15 minutes for the agent to kind of equilibrate in the body. And then we acquire the Cest spectrum for. So what you see here is the final in vivo Cest spectrum on the very right. And very exciting for me was that I did see the change in the amide proton. The amide proton, as you can see, almost disappears. The salicylic acid proton is still there. Great. And I also then generated these pixel-wise parametric maps to look at the Cest effect for the salicylic acid and the amide proton. I then used these pixel-wise parametric maps, or the quantitative numbers, to put it into this equation of reaction coordinate. And this reaction coordinate is basically your enzyme activity, right? Because what you're doing is you're doing 1 minus the amide signal over the salicylic acid signal, which is your control, which gives me these activity maps that shows very high enzyme activity, which then told me that, okay, I might be able to then detect enzyme activity with this agent in vivo in the mouse, which is great. So then I did some control studies. So for the control studies, I generated an ovarian cancer 3-tumor model, orthotopic, which had a medium to low expression of the enzyme. And in 2 of the mice, I injected the agent into the muscle, where you're not expecting any enzyme activity. So then if you look at the Cest spectrum on the very right, you see that what I expected is what I see. You can see that the amide proton signal remains for both the OVCAR3 and the muscle. You don't see enzyme activity, and that's the first validation that the loss of the amide signal that I saw for OVCAR8 was indeed due to enzyme activity and not just the agent getting cleaved or disintegrating in the body. What's very interesting is also so this picture on the figure on the very left is not very important. It is just I was talking about generating pixel-wise parametric maps to just give you a visual idea about what it means. Is these are pixelated parametric maps. What it shows you here is where the agent is. So when I generate the pixelated pixel-wise parametric maps, I can see where the agent localized in the tumors, basically, which is a good sign to know. But for any translational studies, it is important to actually look at the pharmacokinetics of the agent. Is it being washed out of the tumor, or does it stay in the body for a longer time or not? So to do that, I did a temporal analysis of both the peaks at 9.5 ppm, 9.2 ppm for salicylic acid, and the 4.8 ppm peak for the amide proton. And you can see here that within the time frame of the MR experiment, both signals have decreased or have started to decrease, which is a good sign, which means that the agent is getting washed out of the tumor pretty within the time frame of the Cest MR experiment. But if you take a ratio of the decrease of the amide signal versus the salicylic acid signal, you can see that the amide signal decreases much faster compared to the salicylic acid proton, which is another validation that the decrease in the amide proton signal is indeed due to enzyme activity. So with that, I just wanted to give you a summary of all the other studies too. So in summary, during my PhD, I was able to design this common platform of non-metallic Cest MRI agents for detecting enzyme activity within in vivo tumor models for the first time. And this in vivo enzyme detection with Cest MRI method could potentially differentiate aggressive versus a non-aggressive tumor, as I showed with the OVCAR8 versus the OVCAR3. Optimization of the method could be used for planning to pre-screen patients for optical surgical navigation. And with some of the other projects, I also studied enzyme inhibition. So enzyme inhibition studies with Cest MRI methods could assess early response to chemotherapy drugs. Most chemotherapy drugs are based on enzyme inhibition, so this could be a non-invasive method to see the response. And with Cest MRI methods, I was also able to detect an extracellular enzyme activity and inhibition that could be then correlated to a change in an intracellular signaling pathway, such as the NF-kappa beta. Okay. So with that, I would like to move on to a synopsis of my postdoctoral research at the NIH. So when I moved to the NIH, I kind of shifted gears a little bit from cancer imaging to infectious diseases imaging. And I mainly focused on looking at the neuropathology of these diseases with mostly positron emission tomography or PET imaging and some MRI. And some of the biomarkers, and actually before that, just to kind of give a layout, most of the infectious diseases studies I did was with HIV, and some were with cerebral malaria and Ebola, Marburg. So this is just corresponding to the HIV research. These are some of the biomarkers that I evaluated, which is, we looked at the process of neuroinflammation in the brain during the time course of the disease at various stages, starting from infection to treatment and throughout the life course or disease course of the animal using an SIV rhesus macaque. I would actually, I'm going to show a little bit more details on this project later on in the presentation. Then in the same macaque model, we also evaluated the metabolic activity or changes in the metabolic pathway, metabolic dysfunction in these animals using fluorodeoxyglucose, 18F-FDG fluorodeoxyglucose as a marker, and how and then analyzed how that metabolic activity correlates to immune dysfunction in these animals when they are on antiretroviral therapy or they are off antiretroviral therapy in both the brain as well as in the periphery. We performed some MR volumetric studies to look at the changes in different brain components in the brain, to look at the volume changes in different brain components, and how those volume changes can correlate to a neurobehavioral impairment in HIV transgenic rat and how it compares to patients. Most interestingly for me was to look at the dysfunction of systems, where I focused on looking at serotonin, the upregulation of serotonin transporters in SIV infected macaques using another PET tracer, carbon-11 labeled as, and the dopaminergic dysregulation. We looked at both pre and the post-synaptic dysfunction with 2 PET tracers. So I used fluorinating labeled falipride, which targets the post-synaptic D2, D3 receptors, and with 18F FPCMT, which is a carbon-methoxy-nortropin ligand, we targeted the presynaptic dopamine transporter dysregulation. So, okay, apart from HIV, it's very interesting to actually also look at cerebral malaria. And so in case of cerebral malaria, you know, it is one of the deadliest complication of Plasmodium falciparum infection, against which currently there are no anti-malarial drug which is effective. So in order to pave the way for new therapeutic drugs, it is important to understand the underlying disease mechanism. And to do that, we developed this preclinical model of experimental cerebral malaria. And first of all, we were able to see the same symptoms such as edema, brain, blood-brain barrier disruption, brain hemorrhaging in these animals, as is often seen in patients. And in these patients, what is scary about the disease is that by the time that clinical symptoms starts to show up, which is by day 5 or day 6 or post day 5 in the infection, and the patients are admitted within 24 to 48 hours, they actually succumb to the disease. So you need to find an effective method in which by day 4 or by day 5, you can identify the symptom or you can identify the disease. So we used a glutamine antagonist that we developed along collaboratively with Johns Hopkins University. And using this glutamine antagonist injected into the mouse, we were actually able to show the reversal of these same symptoms as early as day 7. And I just wanted to show a snapshot of what I'm talking about here. These are longitudinal T2 weighted MR images, and longitudinally collected over this time frame. And what you see on the first panel here on the very top is the olfactory bulbs where most of the disease starts. As you can see here on day 6, you can actually clearly see the edema with these hyper intense streaks. And on day 5, we have already registered the drug. On day 6, the disease has still progressed to a certain level that you can see the edema still, but by day 8, you can see the reversal. You don't see the edema anymore. This is in the same animal. You don't see the edema anymore, and we followed it up till day 14, beyond which the animal survived. So this was very exciting because these results support the premise that a glutamine antagonist could pose a new direction for adjunctive therapies for cerebral malaria. And then in another side-alone study, I just wanted to show a snapshot of this because this is not directed towards any particular infectious diseases, but basically I was trying to look at the appearance of inflammation, neuroinflammation, or a sterile inflammatory response upon pulsed focus ultrasound treatment in the brain. So as many of you may know, pulsed focus ultrasound with microbubbles is currently one of the most advocated methods for transiently opening the blood-brain barrier for more efficient delivery of neurotherapeutics into the brain. Because most drugs otherwise cannot cross the blood-brain barrier and be treated, right? But with this study, I used MR imaging and PET imaging. With PET imaging, you can see that with the PET tracer, there is upregulation or more intense signals showing that more of the tracer in these regions corresponds to more neuroinflammation in these regions upon multiple pulse focus ultrasound treatments. If you look at the ex vivo MR images, you can see there's hypointense signals in these regions where pulse focus ultrasound was applied. These microintense signals actually correspond to microhemorrhages in the brain. So this study advocates for to look at this method of treatment more appropriately before we move on to the clinical translation of this pulse focus ultrasound technology. And just a little bit about the basics of PET imaging before moving on to one of my projects. So what PET imaging or positron emission tomography involves, it is another non-invasive in vivo imaging technique. It uses a relatively short-lived radio tracer such as fluorine-18, carbon-11, and others, either in their pure form or as part of a larger molecule, as you can see here, that can be synthesized with the radioisotope incorporated within the structure. Once the radio tracer is injected and the subject is placed inside a PET scanner, what happens is this radio tracer emits a positron, which travels a short distance that is which travels a short distance, which is dictated by the energy with which it is emitted. And at a certain point, it meets an electron. When a positron meets an electron, they destroy each other by a process called as annihilation. And this whole process converts the total mass of this electron and positron to energy in the form of two 511 kiloelectron volts of photons that travel 180 degree opposite from each other. Detection of these photons are then achieved by this ring of detectors that you have, and you get a sinogram basically. And all of this coincidence processing can then be fit into an algorithm that has been written or that comes with your PET scanner. And once you fit this raw data into this algorithm and construct the image, you get a PET image like this show up on your computer. So where does chemists play a role in this? So right now there is development of novel PET tracers is very a rapidly growing field for oncology and for neurodegenerative diseases as well. That is because fluorine-18 FDG is the most popular. It's the only FDA approved radio tracer, but it is not a great radio tracer because it is not tumor specific. There is the need for more disease specific radio tracers, and currently there is some progress that is being made. For example, we have amino acid based PET radio tracers such as fluoroethoxytyrosine and F-DOPA that have shown more specificity to certain tumor cells compared to FDG because they are targeting protein synthesis and not glucose metabolism. Then for imaging tumor cell proliferation, there is thymidine-based radio tracers that can differentiate a tumor from a neighboring inflammatory region, which is very important again in the clinic. And there is also tracers that are being developed for imaging tumor hypoxia. They are mainly nitroimidazole-based tracers. This nitro group gets reduced in a hypoxic environment and gives you that signal after getting trapped. But the most important, or I shouldn't say important, the most growing field right now is actually in the area of immunopet, because it holds so much promise for current immunotherapy techniques. It's where you have radioactive metals that can be bound to radioactive metals like copper 64 or zirconium, gallium, that you can bind to antibodies and peptides by conjugating them to these kind of metal chelators, and then this can track specific immune cells or proteins inside of interest in the body, which can be a diagnostic tool to actually see the efficacy of immunotherapy procedures. Okay, so moving on to one of the projects that I did at the NIH. This slide is just to give you an introduction about the pathophysiology of how the CNS is damaged or infected in the HIV, and showing some of the major pathways or factors contributing to that CNS damage. So HIV enters into the brain through infected monocytes, and then it infects other cells such as macrophages and microglial cells and astrocytes. Neurons, they typically don't get infected by the virus. However, there is a multitude of factors that contribute to this neuronal loss, such as the loss of the supportive role of astrocytes, the neurotoxicity from the viral proteins that are released, and most importantly, neuroinflammation, where resting microglial cells get activated, releases a bunch of pro-inflammatory chemokines and cytokines, eventually causing damage to the brain or the neurons and causing neuronal injury or cell death. So what happens, because I am going to talk about the neuroinflammatory aspect, what happens during a neuroinflammation or a CNS injury to the brain is that when resting microglial cells get activated, they change their morphology to this kind of amoeboid kind of morphology. But also importantly, there is an increase in the number of mitochondria and this external mitochondrial protein, which is a peripheral benzodiazepine receptor, or now called as the translocator protein. So you can use this increase in the number of translocator protein or increased expression of this TSPO protein as a surrogate marker to account for neuroinflammation. So, and that is currently actually one of the only biomarkers for looking at neuroinflammation. Therefore, a lot of research, a lot of development has been made towards making that perfect TSPO targeting ligand. The first one that was made was this PK1195, which obviously did not succeed for various reasons. First of all, it has carbon-11 labeling, which is very short-lived radio tracer. But most importantly, this had a very low brain uptake and extensive binding to plasma proteins. So this is what has advanced since then and continues to advance. So there is a great interest in this area where people are trying to make that perfect ligand by various substitutions, making second generation TSPO ligands with better specific to non-specific binding in the brain, improved affinity for TSPO, but still it has not succeeded to be in the clinic yet, because mostly because there is this TSPO polymorphism of the TSPO gene involved, which means that 10% of the patients actually show no binding to most second generation ligands. There are also other disadvantages, like TSPO quantification is very difficult. You want something to go to the clinic, you want a simple tracer, you want a tracer that you inject into a patient. The clinician opens the PET scan, draws an ROI in the brain or draws a region of interest in the tumor, gets a value, knows what it is. As I will show you later on, the whole analysis of these TSPO PET ligands is so complicated that it poses a challenge for clinical translation as well. So this is the synthesis of the TSPO ligand, and the precursor is very simple to synthesize. You basically tosylate this, and then you do very standard radio labeling procedures to get fluorine 18 labeled DPA714 tracer. And basically, the aim of this project came from this hypothesis. We were doing this clinical study with HIV patients that had been under antiretroviral therapy for a very long time. So they had effective antiretroviral treatment, their viral loads were subdued, yet these patients with effective, successful antiretroviral therapy are more prone, or they are at a more risk of getting neurocognitive dysfunction, neurocognitive impairment. So to understand whether there could be a possible early irreversible injury associated with neuroinflammation, we decided to study non-invasively the changes in the brain and the changes in the neuroinflammatory aspect in the brain from the early stages or acute stages where the viral load is very high. Now you have to understand that there is also a paucity of tissue samples or data that is available at early stage in a disease than at a later stage of the disease when the disease has been identified, or the HIV has the patient has been identified to have that disease. So going with that, we developed a rhesus macaque model with a neurotropic strain of SIV. And this neurotropic strain developed SIV encephalitis in 80% of susceptible animals with very high CSF viral load at early stages in the disease. The biomarker I was targeting was translocator protein. I performed longitudinal PET imaging studies with one second-generation Tspo ligand, which was this fluorinating DPS714 ligand. And basically, by longitudinal PET imaging, I mean that the animals were imaged first at baseline when they did not have the SIV strain, and then at multiple time points after inoculation of the SIV strain, after they were being treated. And then finally, we correlated the imaging results to immunohistochemical staining results that we had to get a complete picture of what was going on. So this is a dynamic PET imaging representative figure to just kind of show you the time activity curves and how it looks like. So basically, we put the animal is put inside the scanner, just like a human would go inside a PET scanner, and you inject the fluorinating labeled radio tracer. And this graph here, what I'm showing is a time activity curve. Basically, it is giving you an idea about the uptake of the ligand and the washout of the ligand during the time frame of the experiment. So as you can see here, this is the total binding of the DPS714, and we had to displace because, again, of analysis issues. So I should give a little bit of introduction here. So basically, you can image or you can analyze this PET data very easily if you have a reference region, or most PET ligands, they bind to a specific receptor or a specific protein, right? And usually the specific protein or receptor is localized in a certain region of the brain. For example, if you have a dopamine receptor, it is localized in the midbrain. So you have cerebellum as a reference region that you can then use to do a pharmacokinetic modeling where you have a reference tissue model. But in this case, as you can see in this figure, there is no reference region. The ligand is taken up everywhere in the brain in the same way. So in this case, you could do an arterial input function where you would take a region of interest in the artery where there is no uptake of the ligand as a reference. In these animals, arterial input function, doing that was very difficult. So what we did instead is after 45 minutes into the scan, we displaced with cold PK11195. Now, if you remember, PK11195 is is an antagonist of TSPO. So it can displace the DPS714 here, the bound DPS714, and what you get here instead is non-specific binding. So I kind of used a very complicated analysis where I calculated the specific binding of this ligand, DPS714, to the TSPO by subtracting this unspecific binding from the total binding. And you can see that here, these are my parametric maps. This is the map for total binding of the ligand in the brain. And this is the map from the non-specific binding. If I subtract this from this, I get the specific binding of the TSPO ligand DPS714. So that was just about a little bit about the analysis of the data. But if you look at the imaging results, we saw something that was very, very surprising and completely opposite to what we were expecting, which often happens in research, right? So what we were expecting is, at acute stages of the disease where you have very high CSF viral loads, you are expecting a lot of microglial activation. I was expecting a lot of neuroinflammation. But if you look at the images, it is completely the opposite that I saw. This is the baseline PET scan, and you can see the uptake of the ligand here. And this is an acute stage in the disease when the viral load is 10 to the power 6 copies per mil. You see a decreased binding. And this is not like something that I observed at one time point. If you look at the figure on the right, I have DPS714 binding potential that I calculated on the left, and this axis shows the CSF viral load, right? This dashed line here is when the animal was inoculated. So after inoculation, you can see that within 4 weeks, it reached acute stage, viral load shoots up. When the viral load shoots up, you see that there is a decrease in the binding as opposed to increase in DPA binding. And then we start treating these animals with antiretroviral drugs. When I start treating with antiretroviral drugs, at this time point 3 to 4, you see that viral load now starts to go down. When viral load is going down, I see the same inverse relation that DPA-714 binding increases. We stop treatment, viral load again shoots up, and as viral load shoots up, DPA-714 binding goes down. So I did not know what was going on here. And then I looked at the other cohort of animals that I called slow responder. The reason we called them slow responder is even they were inoculated with the same strain. As you can see here, they did not develop the acute stage of the disease till 80 weeks into the inoculation, therefore called slow responders. But even in the slow responding animals, I observed the same inverse correlation of viral load and DPA-714 binding. And this was very interesting, and I could not understand it just based on imaging results. When I did a statistical analysis, this inverse correlation came out to be significant between the binding and the viral load. Then we looked at immunohistochemistry results. And very interestingly, when we look at the immunohistochemistry results, we see that so we stained for IBA1, which is a microglial activation marker. And in the middle panel, we have caspase, which is an apoptosis marker, right? You see that in this in the animals which have a very high viral load, there is not a great microglial activation going on. If you look at the animals with medium viral loads, like when they were being treated, for example, when they reached 10 to the power 4 copies, then you kind of see what you would expect in a microglial activation or a neuroinflammatory sequel. But when I look at apoptosis, I see more apoptosis in the animals with a high viral load at the early stages. Even more interesting is when we study the neuronal marker NUN and astrocytes, we see that there is a co-localization of this apoptotic marker with neuronal and with the IBA1, which was a microglial marker. And what this co-localization of apoptosis marker with these other markers are telling us is suggesting that there is, instead of a glial activation, there is a glial dysfunction when the viral load is that high. So that was very interesting. With what it told us is with very high CSF viral loads, there is a direct infection of the microglia or macrophages that results in apoptotic changes of the glial cells rather than a microglial activation, which causes the decreased Tspo binding. With the lower CSF viral loads, there is more of a neuroinflammatory visual that we get with increased Tspo binding. So this neuronal apoptosis at such an early stage in the disease, we were observing this for the first time, and what this could partially explain would be that irreversible early CNS damage in HIV-infected patients. So if there is a lot of loss of neuronal cells in the early stages of the disease, no matter if you suppress the viral load after treatment, you don't recover those neurons, and that's an irreversible injury from which the patients can never recover. So our data provides new insights into this neuropathology of HIV disease in the acute stages, and it supports early treatment, enrollment on diagnosis. And with that, actually, I would like to summarize this talk by acknowledging all my previous groups at Arizona, where I did my PhD advisor, Dr. Marty Pagel, and the entire group at Arizona. At the NIH, I would like to thank all my PIs and the entire group for their support and training, and all of you for your attention and patience. And with that, I would like to take any questions. Thank you.
Thank you, Sanghita. It was a very wonderful and lucid presentation. Thanks a lot for sharing your work with us. Now, with permission from our principal, madam, can we move on to the question and answer sections, ma'am?
Thank you.
I would request the audience to type in the chat box your questions, or you can unmute your mic and ask. ask the speaker.
Sure.
If you have any questions, you can unmute your mic and ask the speaker, or you can type in the chat box. We will answer your questions. So we can see there's a lot of appreciation. I can find myself in the chat box. So as an organizer, I'm thankful to all the audience and obviously to Sanghita. So if you have any questions, so the speaker is available. We will not get hold of her very soon. So let's ask her some questions. If you have any questions later also, you can just mail us, and I know that the speaker will definitely answer. But if you have anything to ask, you can please proceed. I would request our principal madam. Ma'am, would you please share your feedback? Hello. I would request our principal madam, Dr. Shoma Ghosh, to share her feedback. Ma'am, are you there? Hello, ma'am. Maybe there are some network issues. I'll try to contact her.
Okay.
Hello, ma'am. Can you hear me? Maybe there are some technical issues. Meanwhile, we have the audience, many audience. So if anyone would like to share their feedback, I can see there is our head of the department from the Department of Zoology, Dr. Indrajit Biswas. Indrajit, if you like to share your feedback. Hello, Indrajit, if you would like to share your feedback with us. Am I audible?
Yes, you are. But I think a lot of
there is
feedback in the chat, maybe. So
yes, I know. Oh, okay, that is okay. Okay, I'll just before formally just thanking Sanghita and getting over with today's webinar, I would call our principal ma'am once.
I'm just. I want to thank all of you for the feedback. I don't know if I do see one question.
Yeah, I also see one question. So, meanwhile, can you answer it?
Is there any gene that is responsible? That's actually a very interesting question, and thank you for asking that. But actually, that is not something that I have studied, so I don't want to comment without knowing. If there is a gene that is responsible for it, so to look at gene expression, some of MRI is useful, but the most or to look at which genes are responsible, people have actually used PET imaging because it is more sensitive in that way than MRI to look at the changed expression of genes in tumor models. Or I don't know if clinical studies have been done, but in preclinical studies, they'll usually knock out a gene, and if they find a gene that is responsible, they'll usually knock out a gene and then do imaging on that to actually see the effect. With most animal models, some of these studies that are very interesting and show the roles of these various genes, the importance of them, when you try to extrapolate that to humans, it is not always a linear extrapolation. So that is where the main disadvantage shows up. I see something in a preclinical mouse model, but as we know, it is not exactly the similar situation in humans. So there is a gap still that needs to be filled out in that regard. Thank you.
Ma'am, can you hear me? Hello? I would request Atri or Pooja to contact Principal Madam once. I am trying to contact her, but maybe there is some issues. So before we technically end the session, I would request you to contact ma'am once.
Yes, Didi.
Okay, okay.
She's the best.
Okay, so one more question.
I'm going to answer the first one, which says, which is the best for study cancer in mouse model or in culture medium? You mean like in vitro or in vivo? Culture medium would be in vitro, right? So definitely in vivo is a step forward than just studying it in culture medium or in vitro. Because you are replicating the kind of like an exact scenario that you have inside our body, inside the tissue, where a lot of other factors are always playing that you can't replicate in a culture medium, or you can't replicate exactly in a in vitro culture medium. But to do in vivo studies, the first validation that comes is from culture medium studies or in vitro studies. So they are very important, but in vivo definitely is a step ahead, or doing mouse studies is definitely a step ahead. After mouse, we usually then go forward with either doing that study in macaques or to the phase 1 clinical trials in humans, usually. Which anti-malarial drug is superior, which kills all or major stages of malarial parasites? I actually don't know the name of which anti-malarial drug is superior. I know that for the study that we did, all of the malarial drugs, there's a different study that Hopkins was doing where they were actually studying in the same experimental mouse model that we made. They have studied all of the malarial drugs that are in use, and they found that for cerebral malaria, none of them are actually effective in reversing the disease. But for regular malaria, if your question is targeted at, I don't know which one is the best drug. Actually, there are a lot of out there. It's just I haven't looked into that, I would say. I can get back to you, actually, if I can pass on the question. If I find out the answer, I can pass it on to Amrita. And,
For sure. One more question is there. Is there any investigation regarding congenital abnormalities?
This is for the HIV, right? Is this for the HIV study you're asking?
Indrajitda, can you please specify your query? In case of which disease, if you could just type in the chat box your question. Is there any investigation regarding congenital abnormalities specific to which disease you want to know? If you can specify, it would be easy for her to answer.
Sorry, maybe it is a very simple question, and it's just not my. Okay, schizophrenic diseases. Yeah. Okay. Thank you for asking that. So there is the unit. So NIH Institute, which is more focused on neurodegenerative diseases, they have active clinical trials going on in that area for specific like neuro. Cognitive and neurodegeneration diseases. I wasn't part of that institute, so I don't know about the exact trials, but definitely there are for a specific unit for schizophrenia. Yeah. Yes, quinine drugs are effective for cerebral malaria, not, but for general malaria, yes. Is malarial drug effects of malarial drug effects of band 3 protein? Again, I'm not sure about drug effects. I'm not sure about the exact question.
If you have questions later also, I am just giving the audience, sharing with the audience my email ID. You can drop in your questions and I would forward it to Sanghita. She can answer it. So I'm just typing my mail ID. And we are trying to connect with our principal madam. Maybe for some network issues, she is not being able to be in the connection. So if there are no more questions, I would request our bursar and Associate Professor, Department of Mathematics, Dr. Lipika Mollik, to deliver the vote of thanks, and before that obviously share her feedback. And if meanwhile madam can join with us, it will be very nice. Otherwise we would end today's session. So Lipika di, if you would kindly take over.
Principal madam is here and totally.
Oh, thank you. Thank you, ma'am. We were trying to connect for you.
I tried my best to comprehend, but I couldn't. But I enjoyed the presentation because chemistry is not, of course, my cup of tea, but it can be presented so lucidly. I learned it from Dr. Sina Roy. So I'm really thankful for her effort to teach me chemistry.
Thank you. It was my honor. Thanks.
I'm really thankful because I was listening to every word so far, and I was looking at the slides. I couldn't understand everything, but it was so wonderful. And she's a young scholar, and I must say that our college is honored to have you with us.
Thank you so much. It was just so that everyone there knows, it was also very difficult for me or challenging when I started this research. I did not understand MRI or PET, so I felt like a first-year student every year of my graduate school. Just saying it out there.
But today you taught me a lot.
Thank you, ma'am.
And one thing I must say that thank you, Amrita, also. Amrita is a very beloved colleague of mine, and of course an asset of the college. And today I'm proud to have Amrita as my colleague. Thank you, Amrita.
So thank you, ma'am. It's I'm so much honored. Means you always appreciate us, always, and in all formal platforms. So it is really great. We are very lucky to be under your mentorship and guidance and work under you, as I always say. So thank you for always encouraging and inspiring us with your words, and especially to conduct this type of webinars and academic efforts. You are always there with us, we know. So thank you so much, ma'am.
Actually, I enjoy these webinars because I try to learn all these things, because as you know, Amrita, when these type of seminars are organized in the college, there are lots of works I have to do, all administrative type of So I don't get the time to listen to all these things. So it's a privilege. It is a silver lining in this lockdown for me to learn chemistry from Dr. Sinha Roy, from a scholar like Dr. Sinha Roy. So thank you again. It's over to Lipika to give formal vote of thanks.
Thank you. Thank you, ma'am. This is vote of thanks. It is my proud privilege to offer vote of thanks to the resource person, the participants and the organizers. I must begin with giving my heartfelt thanks and gratitude to our principal, Dr. Shoma Ghosh, for giving us too much pressure for organizing such events, which ultimately get converted into enormous pleasure and academic satisfaction. Honorable resource person Dr. Sanghita Sinade has enlightened us with our resourceful deliberations. Really, we perhaps got the chance to swim in the sea of knowledge due to this illuminating lecture. Hope we will be able to hear from Dr. Sanghita Sinade again, as our esteemed speaker has just contributed some pearls from a treasure of knowledge. I must offer my sincere thanks to all the participants, without whose dedicated participation this webinar would not have been possible. I feel extremely delighted to thank the Department of Chemistry students, which under the guidance of young scholar and teacher Dr. Amrita Banerjee has organized this webinar to give us the opportunity to enlighten ourselves. This is to convey my thanks and gratitude to the webinar organizing committee, its convener Sri Pradipta Mukherjee, Teachers Council Secretary Dr. Pradip Das. I, on behalf of the entire college, take this opportunity to convey thanks to Srimati Atri Bhattacharya and Srimati Pooja Das, two of our beloved junior colleagues, for their dedicated service to manage all technicalities related to the webinar. And last but most importantly, Dr. Rupa Sen, our IQAC coordinator, for taking immense effort for organizing this webinar. Thank you all. Hello Amrita, thank you.
Amrita, it's over to Amrita. অমৃতা কেউ কি জানতো যে লিপিকা ম্যাডাম এত ভালো বলতে পারেন? So
এবার তো আমি this is wonderful ma'am. So now এবার আমি সব সময় লিপিকা দিকে I'll catch hold of her and to end the webinars like this. She is always with us in all the webinars, I know, silently, but now on she will take this official responsibility.
It is also এটা আমাদের silver lining in this lockdown, না? লিপিকা ম্যাডাম
Definitely. Definitely. Last webinar I missed her because I informed her in a very short notice and she had some medical issues, so she could not. But from now on, so I would not leave any opportunity to engage Lipika Di in this type of webinars and conveying the vote of thanks and feedback.
We will not allow her to remain behind the screen always.
No, not anymore.
Not anymore.
That is right, under the screen.
So, thank you all, ma'am. Will we end it now?
Yeah. Thank you.
Okay, so thank you everyone.
Thank you all. I'm going to leave the chat. Hope to reconnect with you all at some point again in the future.
Oh, definitely. It will be an honor.
This is an open invitation to you, Dr. Sinha Roy, from all of us.
Thank you. Honor is all mine. I'm very honored actually with everything that you all said, and thank you so much for the invitation once again.